Good day everyone. We are the group 2 and today we'll be tackling the last topic for this semester, which is gene regulation. So by the end of this lesson, you should be able to differentiate between gene regulation in prokaryotic and eukaryotic cells, describe the operant model of gene regulation in prokaryotes, and explain the mechanisms of gene regulation in eukaryotes at multiple levels. But before we start, let us first have a short review in the previous discussion on the central dogma. So the central dogma is a fundamental principle in biology that outlines the flow of genetic information within biological system. It states that genetic information flows from DNA to RNA to protein. So the central dogma has three main processes. We have the DNA replication, transcription, and translation. So when we say replication, the DNA duplicates itself to create identical copies of itself. While in transcription, the RNA polymerase will read a DNA sequence and synthesize a complementary RNA molecule called the messenger RNA or the mRNA. In the translation, the mRNA molecule is translated into protein. So ribosomes read the mRNA sequence in codons, which is a triplets of nucleotides, and match them with corresponding anticodons. So now that we've refreshed our memory on the fundamental principles of the central dogma, we'll now turn our attention to the mechanisms that govern gene expression. Let's first define a few terms so that you will be guided throughout the discussion. So the first term that we're going to define is gene expression. So gene expression is a process of turning on a gene to produce mRNA and protein. So turning on means the process by which a cell uses information in a gene to produce a functional product, which is typically protein. So it involves two main processes in the central dogma, which is the transcription, where the DNA is copied into messenger RNA, and translation, where the mRNA is used as a template to assemble a protein. This process allows cells to carry out specific functions by producing the proteins they need. Not all genes are expressed at all times. Cells only activate the genes necessary for their current tasks. Now, what facilitates this only activation of genes necessary? So this is facilitated by gene regulation. So when we say gene regulation, this is the mechanisms and processes that control the phenotypic expression of genes. So by control, it means gene regulation is responsible to when, where, and how much a gene is expressed. It ensures that genes are turned on or off as needed depending on cell's environment, type, and function. By controlling gene expression, gene regulation ensures that the right proteins are produced at the right time and in the right amounts, enabling cells to respond to changes and maintain proper function without regulation. Gene expression would be random, chaotic, disrupting cellular processes and overall organism health. So gene expression and regulation are closely related to one another. Gene expression refers to how the gene is being expressed, while gene regulation refers to how this gene expression is regulated. So this regulation on gene depends on the coordinated action of different type of genes. So specifically, we have the regulatory genes and structural genes. So when we say regulatory genes, this is the DNA sequence that encodes a protein or RNA molecule that interacts with DNA sequences and affect their transcription or translation or both of those processes. While structural gene is the DNA sequence that encodes a protein that function in metabolism or biosynthesis or that has a structural role in the cell. So in simple terms, when we say regulatory genes, so these are genes that code for proteins that regulate the activity of other genes, while structural genes codes for proteins that perform specific functions in the cell. Let us now move on to how gene is regulated how gene expression is regulated in prokaryotes. Prokaryotes, primarily regulate gene expression at the transcriptional level through operons. So operons are a set of structural genes in bacterial cell along with their common promoter and other sequences such as an operator that control their transcription. A key example of an operon is the lock operon in E. coli. So the lock operon regulates the metabolism of lactose in E. coli bacteria. So maintaining operons consumes energy. Since maintaining active operons consumes energy, prokaryotes are smart about when to activate or when to deactivate them. So the LAC operand consists of several genes, including the LAC-Z, LAC-Y, and LAC-A. With these genes encodes for specific enzyme necessary for the digestion of lactose. So in the presence of lactose, the LAC operand is activated to produce these enzymes. The process begins when the lactose enters the cell and binds to the repressor protein. So this repressor protein without lactose is nakastick niya siya together with the operon. Once there is a presence of lactose, it will bind to the repressor protein, causing it to change shape and detach from the operator, allowing the RNA polymerase to bind to the promoter and begin the transcription of the LAC-Z, LAC-Y, LAC-A enzymes. So if the lactose is absent, the repressor will bind to the operator once again, preventing RNA polymerase to code these following enzymes. By doing this, bacteria helps conserve energy by not producing unnecessary enzymes. The lock operon is an example of inducible gene regulation where the presence of an inducer, which is lactose, activates the expression of specific genes. So that ends my part for how gene expression is regulated in prokaryotes.
So, good day everyone, and today we will tackle about the eukaryotic cells and bacteria, where it is a tale of shared and divergent gene regulation. So, this topic explores the fascinating processes that regulate gene expression in two major domains of life, where it is the eukaryotes and bacteria. And as the report or my as I go deeper or tackle about this topic, we will examine their similarities, differences, and what does this mean for our daily understanding about the eukaryotes and bacteria. So first, we have here the unveiling the fundamentals of gene regulation, where it has eukaryotes and bacteria. So gene regulation in eukaryotes involves a complex interplay of transcription factors, chromatin structure, and post-transcriptional modification. So gene regulation is crucial for the proper functioning of all living organisms. It determines when, where, and how much of a specific gene is expressed. While the eukaryotes, gene regulation is incredibly complex due to their multicellular nature and diverse cell types, which this involves the transcription factors that turn genes on or off, while the organization of DNA into chromatin, which can either block or promotes access to genes, and the post-transcriptional modification, which fine-tune gene generation after transcription. So, mo to'y maka-explain about sa eukaryotes, while the bacteria is it utilize a more streamlined system relying heavily on the interplay of transcription factors, sigma factors, and small RNAs to control gene regulation. So this means that bacteria, being uncellular and simpler organisms, have a streamlined system which they rely on transcription factors, pareha sa eukaryotes, the sigma factors which help RNA polymerase recognize promoter regions, while the small RNAs play a regulatory role in gene expression. So that's explained the eukaryotes and the bacteria. Next, let's deeper to the similarities in the language of transcription. We have here three similarities: the transcription factors, the promoters, and the RNA polymerase. So first, I can, despite these three differences, both eukaryotes and bacteria share gihapon og fundamental elements nga magpareha silang gene regulation process. First, the transcription factor. So sa transcription factors, both eukaryotes and bacteria employ transcription factors to bind to specific DNA sequences regulating the initiation of transcription, which means the proteins bind to specific DNA sequences to regulate transcription. Both domains use transcription factors to activate or suppress the expression of genes, while the promoter regions, starting points for transcription, are present in both domains, providing a platform for RNA polymerase binding. So this kanang ang mga DNA regions where transcription begins. So dili magsugod as promoters, kaya ang promoters serve as binding sites for RNA polymerase, helping to initiate gene expression in both eukaryotes and bacteria, while the RNA polymerase both utilize RNA to synthesize. It is a fundamental step in gene expression. So this enzyme is responsible for synthesizing the mRNA from DNA. So mo ni siya kanang gene expression shared across life forms. So these three similarities highlights how these processes are conserved through the evolution of the transcription of the three factors. So next is constructing the landscape of chromatin structure. So kuan siya kanang murag, so itaw gani kanang structure sa point which is a major point of divergence. We have here the first kay eukaryotic chromatin, next nucleosomes, then epigenetics. So let's break down this. And first kay eukaryotic chromatin. So in this point, in eukaryotes, DNA is wrapped around proteins called histones to form structure called nucleosomes. This creates a higher level of organization known as chromatin. So chromatin can be modified to either promote or repress transcription. While kaning mga DNA, epigenetics, both ni sila structure para mahimo ang chromatin. While the DNA methylation adds methyl groups to DNA, often silencing genes, and the histone acetylation makes chromatin more open, enhancing gene expression. So about na siya sa eukaryotic chromatin, and also the DNA is tightly wrapped around histone proteins, forming nucleosome, the basic unit of chromatin structure, while the epigenetics eukaryotic genomes employ a diverse range of epigenetic modifications, including DNA methylation and histone acetylation to regulate gene expression. And their DNA is less organized, so while allowing this to access to genes, instead they regulate gene expression mainly by controlling transcription initiation through this structure. Next is the Next is the post-transcriptional regulation, a shared dance with unique steps. So post-transcriptional regulation refers to how cells control mRNA after it is transcribed. Here, we see both shared and unique aspects. While in eukaryotes, mRNA undergoes extensive modification. Splicing removes introns and joins exons to create a functional mRNA, and capping adds a protective cap to, to the 5' and polydenylation adds a tail of adenine nucleotides. So these processes ensure that the mRNA is stable, can leave the nucleus, and ready for translation. The RNA processing, the RNA degradation, and the small RNAs. So in bacteria, transcription and translation happen simultaneously. So these modifications are not necessary. However, both means regulate mRNA stability using small RNAs which bind to mRNA and either stabilize or degrade it. Next is the translational control, a symphony of conserved mechanisms and divert orchestration. We have here the ribosomes, the tRNA, and the initiation factors. So translation is the process where mRNA is decoded to build proteins. Here, we can find both conserved mechanisms and unique differences sa ilahang tulo. Ribosomes kay both domains utilize to remain the mRNA into proteins ay to translate them with a conserved core structure and functions. So both domains daw have ribosomes which are molecular machines for protein synthesis. While the basic structure is conserved, eukaryotic ribosomes are larger and more complex, while the tRNA transfer RNAs are universal. They bring amino acids to the ribosome, matching amino acids, while the initiation factor states that eukaryotic translation involves more initiation factors than bacteria. This added complexity gives eukaryotes greater control over when and how proteins are made. Next is the navigating the labyrinth of signal transduction and regulatory networks. Here we only have eukaryotes and bacteria. So gene regulation is also influenced by signal transduction pathways, the way cell responds to external signals. Where in the eukaryotes, these pathways involves complex cascades of protein interactions and modification. For example, hormones can trigger changes in gene expression through these pathways. While the bacteria, this is the systems that are simpler but very efficient. For instance, when nutrients are scarce, bacteria can quickly activate genes to adapt to the new condition. So, ang eukaryotes ug ang bacteria kay na siya mga difference silang duha. Next, is the understanding cellular complexity, the insights from the interplay of eukaryotic and bacterial gene regulation. So, by understanding the similarities and differences in gene regulation between these two domains, we gain valuable insights into the evolution and complexity of life. In conclusion, by comparing gene regulation in eukaryotes and bacteria, we gain insights into the evolution of life. The similarities remind us sa kanaang unsa jud about ang two, the bacteria and the eukaryotes. So, understanding these processes help us tackle challenges sa mga gene therapy o mga synthetic biology. So that is all about my report about the eukaryotic cells and bacteria. Next, we will tackle about the dynamic dance of chromatin, how structure impacts gene expression. In this presentation, we will explore the relationship between chromatin structure and gene regulation in eukaryotic cells, where the chromatin is the complex of DNA and proteins that makes up chromosomes. Its structure is not just for packaging, it plays an active role in determining which genes are expressed or silenced. So the first is the chromatin structure and the regulation, the building block and the regulation, where eukaryotic DNA is incredibly long and must be efficiently packaged to fit inside the nucleus. This is achieved through the chromatin, which consists of DNA wrapped around histone proteins to form nucleosomes. So the basic units of chromatin, mo to ang basic units of chromatin. So the structure of chromatin is not static. It can dynamically change. So these changes regulate the accessibility of genes to the transcription machinery, which ultimately affects gene expression. So mo na ang the building block and the regulation. Next is the histone modification and chromatin. So, chromatin remodeling. Histone proteins in nucleosomes are subjects to various chemical modifications that influence chromatin structure and gene expression. Specifically in the acetylation, methylation, and remodeling. Acetylation, adding acetyl groups to histone tails, loosens the chromatin structure, making genes more accessible to transcription factors and RNA polymerase, thereby increasing transcription. So that's how this is the effects of histone methylation depends on the specific site. It can either be activated or repressed through gene expression. While the remodeling specialized enzymes reposition nucleosomes, exposing or hiding DNA regions, further influencing transcriptional activity. This is a dynamic and energy-dependent process. So that's how explains the modifications of chromatin, the acetylation, methylation, and remodeling. Next, we have the DNA methylation and gene expression. We have here the expression, repression, and the regulation. So DNA methylation is another key mechanism in gene regulation. Nag-involve ni og mga methyl groups to cytosine bases, which especially in CPG islands, which are regions rich in cytosine and guanine nucleotides. Methylation, on the other hand, usually silences genes by preventing transcription factors from accessing the DNA. While the DNA methylation patterns can change during development or in response to environmental signals, making it a flexible and crucial regulator of gene expression. So, mo to ang mag-explain sa repression and the regulation of the DNA. Next is the transcription factors and chromatin dynamics. We have here the transcription factors can recruit chromatin, remodeling complexes, altering chromatin structure, and promoting gene expression. And second, they can also bind to specific DNA sequences, recruiting co-activators or co-repressors to influence chromatin accessibility. So first, transcription factors play a dual role in chromatin dynamics. They recruit chromatin remodeling complexes which it either promote or inhibit transcription, and also they bind the specific DNA sequences and to attract that co-activators or co-expression. So this interplay ensures that gene expression is tightly regulated and responsive to the cellular needs. Next is the epigenetic inheritance and development. So epigenetic modifications such as histone changes, the DNA methylation can be passed down to daughter cells during cell division, or kaning mga inheritance is crucial for maintaining cell identity and enabling proper development. Moreover, kaning nga external factors like stress, diet, and toxins can alter epigenetic marks. So through these changes, contribute siya sa developmental plasticity but can also increase susceptibility to disease. So mo to ang importance sa epigenetic inheritance and development. Next is the environmental factors and chromatin's changes through structure gapon. First, the exposure. Second, the modification. Ay first kani exposure kay factors ani kay stress, dietary habits or toxins that can induce chromatin changes. While the modification, these factors kay leads to epigenetic alterations that can persist long term o para ni mga stress, diet, and toxins. Last is the phenotype. These changes manifest in physical traits and can increase the risk of disease or alter developmental outcomes. So these changes daw can contribute to diseases and ma-develop siya into developmental outcomes. Last, sa implications for human health and disease, where understanding chromatin structure has profound implication for human health. Mao ni siya first, cancer. Second, neurological disorders. Third is metabolic disease. The cancers is the abnormal chromatin modification that can activate oncogenes or silence tumor that nga ma-form into cancer. Next is the neurological disorder are linked to conditions like autism and Alzheimer's disease. So mao ni mga disorder nga pwede magkuha sa mao ni ang mga neurological disorder. Next is the metabolic disease, which dysregulation of epigenetic and processes contributes to disease such as diabetes and obesity. Mao na siyang importance implications of for human health and disease. So by studying chromatin, we open doors to targeted therapies that can reverse these harmful challenges. So in all, chromatin is more than a structural entity. It actively regulates gene expression and responds dynamically to internal and external signals. So this interplay between chromatin structure and function is essential for our daily lives. So that's all for my reporting about the eukaryotic cells and bacteria and the dynamic of chromosomes. Thank you for listening.
So the initiation of transcription is regulated by transcription factors and transcriptional regulator proteins. Regulation of transcription initiation in eukaryotes. So the regulation of gene expression in eukaryotic cells, focusing on the initiation of transcription. So it explained that this process involves two key levels of control. The first one is the chromatin and DNA structure alteration. This is one level of control. The second one is the protein binding of DNA sequences. This is another important level of control. Specific proteins bind to DNA sequences, regulatory elements to influence transcription. These proteins include the general transcription regulator proteins, and the transcriptional activator proteins. So the general transcription,
These are part of the basal transcription apparatus, which binds to the core promoter and enables minimal transcription. So the second one is the transcription regulatory proteins. So these proteins bind to regulatory promoters and enhancers, which are located upstream of the core promoter, so they can be activators and repressor. The activators is the stimulate transcription, while the repressor is inhibit transcription. So the last one is transcriptional activator proteins. So these stimulate and stabilize the basal transcription apparatus at the core promoter. They may interact directly with the basal transcription apparatus or through co-activator proteins. Some also have activity altering chromatin structure to stimulate transcription. So this emphasizes the importance of these proteins and regulatory elements in controlling the level of transcription in eukaryotic cells. The basal transcription apparatus alone allows for minimal transcription. The additional regulatory proteins are necessary for normal level of transcription. So next, we have here the mechanism of transcriptional activation in eukaryotes. So the DNA, as you can see in this image, the DNA, the genetic material containing the gene to be transcribed. So the core promoter, this one, kini siya. The core promoter, the region of DNA near the transcription start site that contains the Tata box, it's where the basal transcription apparatus binds. So the regulatory promoter or the activator binding site, kini siya. A region upstream of the core promoter where activator proteins bind. The transcriptional activator proteins. These proteins binds to the specific DNA sequences or consensus sequences within regulatory promoters or enhances. So they have two main functions. The first one is the DNA binding, and the second one is the interaction with other components. So the enhancer, kini siya diri dapit. Enhancer, a DNA sequence that can be located far from the core promoter but still enhances transcription. So the co-activator, kini siya, is a protein that mediates the interaction between activator proteins and the basal transcription apparatus. While the mediator is a large protein complex that bridge the interaction between activator proteins and the RNA polymers. The basal transcription apparatus, a complex of proteins including transcription factors and RNA polymers that binds to the core promoter and initiates transcription. This includes general transcription factors, the RNA polymers, and the transcription start site, the point on the DNA where transcription begins. Sama ni siya dere. So the next one we have here, the next one is the principle of mix and match consensus sequence in eukaryotic gene promoters. So the consensus sequences specific short DNA sequences like Tata box, Kaat boxes, and GC boxes and octamer elements found in promoter. So this sequence serves as binding site for transcriptional activator proteins. The promoter variability, there are three different eukaryotic gene promoters: the SV40 early promoter, the thymidine kinase promoter, and the histone H2B promoter. Each promoter contains a unique combination of these consensus sequences. So the combinatory regulatory or the regulation, rather, different transcriptional activator proteins bind to specific consensus sequences. The unique combination of these sequences in each promoter determine which activator proteins can bind, leading to specific diverse gene regulatory. For example, galactose metabolism, example of how this combinatory regulation works in galactose metabolism in yeast. The GAL4 activator protein regulates the transcription of genes involved in galactose metabolism. In summary, this image precise combination of consensus sequences in a promoter determine which transcriptional activator proteins can bind, resulting in a unique regulatory profile for each gene. So this combinatory control allows for a high degree of specificity and diversity in gene expression. So next one, we have here the regulation of galactose metabolism in yeast, specifically focusing on the role of GAL4, GAL80, and GAL3. So the mechanism of it is, first one, the absence of galactose. When galactose is absent, GAL80 binds to GAL4. So this prevents GAL4 from interacting with the UASG or the upstream activating sequence and the basal transcription apparatus. So those inhibiting transcription of the genes responsible for galactose metabolism. So the second one is when the presence of galactose. When galactose is present, it binds to GAL3. This binding causes a conformational change in GAL3, enabling it to interact with GAL80. This interaction causes a conformational change in GAL80, releasing it from GAL4. So the third one is transcription activation. The freed GAL4 is then able to bind to the UASG and interact with the basal transcription apparatus, initiating transcription of the genes needed from galactose metabolism. In summary, GAL80 acts as a repressor, inhibiting GAL4's activity in the absence of galactose. So the presence of galactose triggers a cascade of events involving GAL3 that relieves this repression, allowing GAL4 to activate transcription. So this system ensures that the genes involved in galactose metabolism are only expressed when galactose is available. So the next one is the eukaryotic repressor. So the first one is, unlike bacterial, the transcriptional repressor and eukaryotic cells and their mechanism of action, constructing them with repressor in bacteria. It also touches upon enhancer and insulator. So the first one, unlike bacterial repressor that directly block RNA polymers, eukaryotic repressor utilize different mechanism to inhibit transcription. The second one is competitive binding. Repressor may compete with activators for binding sites on the DNA. The third one is the proximity inhibition. Repressors can bind near activator binding sites, preventing the activator from contacting the basal transcription apparatus. The fourth one is the direct interference. Repressor may directly interfere with the basal transcription apparatus, blocking transcription initiation. So, the function of insulator are DNA sequences that block the action of an enhancer on a promoter when positioned between the enhancer and the promoter. This prevents the enhancer from inappropriately activating a gene that it shows that the enhancer can stimulate transcription of a gene only if it's not blocked by insulator. And that's all for for the for the for the regulation of transcription initiation in eukaryotics.
Eukaryotic gene regulation. Eukaryotic gene regulation, unlike prokaryotes, eukaryotic gene regulation is a complex process that involves multiple levels, including post-transcriptional modifications. These modifications, such as RNA processing and degradation, play a crucial role in controlling gene expression and ultimately determining cellular phenotype. The importance of gene regulation is that gene regulation is essential for various biological processes, including development, cell differentiation, and response to environmental changes. It ensures that genes are turned on or off at the appropriate times and in the correct amounts. Gene regulation through RNA splicing. Eukaryotic genes often contain non-coding sequences called introns interspersed with coding sequences called exons. Before mRNA can be translated, these introns must be removed through a process called splicing. Alternative splicing is a remarkable feature of eukaryotic gene expression is alternative splicing, where a single gene can generate multiple protein isoforms by splicing its pre-mRNA in different ways. This allows for a vast expansion of the proteome. Several factors influence splice site selection, including consensus sequences. So these are specific nucleotide sequences found at the junctions between exons and introns. Next is the exonic or intronic splicing enhancers and silencers. So these sequences can promote or repress the use of particular splice sites. Next is the SR proteins, or the serine and arginine rich. So these proteins bind to pre-mRNA and influence splice site selection. And lastly, the SNRNPs, or the small nuclear ribonucleoproteins. So these complexes are essential for the splicing process. Alternative splicing in T antigen gene. So the SV40 virus provides a well-studied example of alternative splicing. The choice of splice site determines whether the large T or small t antigen is produced. Alternative splicing in sex determination in Drosophila. Alternative splicing plays a crucial role in Drosophila sex determination. The presence or absence of the SXL protein, or the sex lethal protein, determined by the number of X chromosomes, influences the splicing of transformer pre-mRNA. SXL gene and SXL protein. So the SXL, or the sex lethal gene, is activated in females, producing the SXL protein. In males, the SXL gene is inactive and no SXL protein is produced. Influence on TRA pre-mRNA, or transformer pre-mRNA. In females, the SXL protein binds to transformer pre-mRNA, causing it to be spliced at a downstream site. This results in the production of a functional transformer protein. In males, the TRA pre-mRNA is spliced at an upstream site, leading to the production of a non-functional transformer protein. cascading effect on DSX pre-mRNA or double-sex pre-mRNA. So the TRA protein, so TRA means transformer, further regulates the splicing of DSX pre-mRNA, ultimately determining the sex of the embryo. In females, the TRA protein directs the splicing of double-sex pre-mRNA to produce a female-specific double-sex protein. In males, the absence of TRA or the transformer protein leads to the production of a male-specific DSX protein. Next is the degradation of RNA. The stability of mRNA molecules can vary widely, affecting the amount of protein produced. Factors that influence mRNA stability include the 5 cap. So this modified guanine nucleotide cap protects the mRNA from degradation by exonucleases, enzymes that degrade RNA from the ends. It also aids in the initiation of translation. Next is the poly A tail, a long chain of adenine nucleotides added to the 3 end of the mRNA. It protects the mRNA from degradation and plays a role in export from the nucleus and translation initiation. So the length of the poly A tail can influence mRNA stability. So I forgot to add this one. So lastly is the specific sequences in the mRNA. Certain sequences within the mRNA can signal for degradation. For example, the AU-rich elements are often found in the 3 untranslated region of unstable mRNAs. So these sequences can recruit proteins that promote mRNA decay. So the degradation pathways. So first is the 5 to 3 degradation. So begins with the removal of the 5 cup. So exonucleases then degrade the mRNA from the 5 end to the 3 end. Next is the 3 to 5 degradation. So the poly A tail is shortened by a process called deadenylation. So exonucleases then degrade the mRNA from the 3 end to the 5 end. Next is the internal cleavage. The endonucleases cleave the mRNA at specific internal sites. So the resulting fragments are then degraded by the exonucleases. So P bodies. So cytoplasmic granules that contain proteins involved in mRNA decay. They store and degrade mRNAs, playing a role in regulating gene expression. So P bodies can also store translationally repressed mRNAs, which can be released for translation later. So microRNAs and small interfering RNAs. So small RNA molecules that can target specific mRNAs for degradation. So they bind to complementary sequences in the target mRNA, leading to its degradation or translational repression. So microRNAs and small interfering RNAs play important roles in various cellular processes, including development, differentiation, and response to stress.
17.5 RNA interference is an important mechanism of gene regulation. So, this presentation explores RNA interference, a crucial mechanism of gene regulation. We'll discuss its components, mechanisms, role in development and disease, and its potential applications in genetic engineering and medicine. So what is RNA interference? So RNA interference or RNAi is a biological process where RNA molecules inhibit gene expression by silencing specific mRNAs. So it's a widespread mechanism in eukaryotes and regulates about 30% of human genes. RNA interference has also been harnessed in genetic engineering for precise gene expression control. Now we have here the key components of RNA interference. So, the RNA interference process involves small RNA molecules, the siRNAs and the miRNAs. So these are produced by the enzyme dicer, which cleaves double-stranded RNA into smaller fragments. So these fragments then form a complex called RISC, or the RISC, which binds to target mRNAs to mediate gene silencing. So the mechanisms of RNA silencing. So RNA silencing functions through multiple pathways. So siRNAs guide RISC or RISC to cleave target mRNA. So while the miRNAs inhibit translation by binding imperfectly to mRNA. So some siRNAs induce histone methylation, silencing transcription, and others trigger degradation of unstable mRNAs. Now we have the RNA interference in development and disease. So RNA interference plays a significant role in regulating development. For example, miRNAs like MIR1 control heart development. Abnormal miRNA expression can lead to diseases like cancer or heart defects. So these insights have opened doors for therapeutic applications in medicine. Now we have here the RNA crosstalk. So RNA crosstalk refers to the competition among RNA molecules for miRNA binding sites. So non-coding RNAs like the INC RNAs and circular RNAs will act as a decoy, regulating gene expression by binding miRNAs. So this interplay adds another layer of a complexity to gene regulation. So now let us move on to 17.6. The expression of some genes is regulated by processes that affect translation or by modifications of proteins. So this section focuses on how gene expression is regulated at the translational level and through post-translational modifications of proteins. Now we have here the translational control of gene expression. So translation is regulated by the availability of ribosomes and initiation factors. So for instance, immune cells like the T cells rapidly increase protein synthesis during activation. So heat shock proteins are also another example, produced in response to stress to protect cells. Now we have here the post-translational modifications. So once proteins are synthesized, they undergo chemical modifications that alter their function, stability, and also their localization. So examples include the phosphorylation, which can activate enzymes, or the ubiquitination, which marks proteins for degradation. So these modifications are crucial for precise cellular regulation. And lastly, we have here the applications and future directions. So understanding translational control and post-translational modifications provides a valuable insights for medical treatments, biotechnology, and also the synthetic biology. However, challenges like the ethical considerations and the complexity of these processes must be addressed to maximize their potential. So that's it for our report. So thank you and goodbye.